So we're going to basically do 3 experiments in here. We're going to look at the diffusion of 3 dyes in a solid. We're going to look at osmosis, which is the movement of water. Very good. And we are going to look at osmosis, movement of water in plant cells. So this lab is mostly about the graphing at the end. You're going to gather data and then graph it. These are the 3 things we're doing. What I would recommend, because this lab can run long, is that you get the first one started, have it running, because it has to sit for 30 minutes. You get it running while you set up the other 2. What we're going to do for this first experiment, you are going to make 3 little bags out of dialysis tubing. If you run out for some reason, if they didn't put enough on your tray, there's a container here, you can get another piece. It looks like a cellophane ribbon, but it's not a ribbon, it's a tube that's flattened out and then dried out, and the sides stick together. The way you hydrate it is get it wet and rub it, and if you do that enough, the sides will come apart, and then you can make the little bags. Would you agree? I just made a bag and filled it with water.
Okay.
Yeah. You are going to do almost the same thing. I would use tap water to hydrate these. In a lab, you would use this special water, the deionized water, for everything. During this experiment, you're going to make 3 of these bags. You're going to put them in 3 beakers. I want you to have deionized water in the beaker. In one bag, you're going to put 10 mils of the deionized water. In another bag, you're going to put syrup. This is watered down corn syrup, basically. And that's what you're going to use as syrup. So in the one where you put 10 milliliters of syrup, you're going to put 10 mils of syrup. No surprises. You'll measure your 10 mils with your graduated cylinder, and if you have to adjust the amount, you can use this transfer pipette. You have disposable transfer pipettes. They're basically like disposable eyedroppers. You can add a little bit until you get the perfect amount. So 10 mils of water, 10 mils of syrup. You understand those 2 bags. This one, the one that on your graph will be the 50%, it'll have 5 mils of water, 5 mils of syrup. That shouldn't be scary. Are you with me? Mm-hmm. All right, I've got my 10 mils of water. And you might find it hard to pour the water in to start with, so you can then use your transfer pipette. All right, so you got your 10 mils, and then you need to tie it. You see, I went with a knot at the bottom, and I usually go with a knot at the top. Okay, that's how you're going to do your 3 bags. I would get all 3 bags ready. I would tie them shut. The way you do that is you twist it shut. And then you take that long twisty part at the top and you tie it. Now, some people have trouble with making the knot, and for them there are twist ties. You can use twist ties, but you would agree, knotted baggie, right? What you're going to do, you're going to dry your bags off. There's a scale at the back. You're going to weigh them all on the scale. Remember, you have to tare it between each one. That means set it to 0 for each one. But you don't want to weigh it like this where it's covered with water. You want to dry it off first, because what you're going to be measuring is the original weight and the weight after 30 minutes. And then you'll subtract them to get the weight change. What you're going to graph is the weight change. You can understand, if you have drops on it here that are not on the outside there, it messes it up. If you have drops here that you didn't have there, it messes it up. Dry it really good both times. Are you with me?
Yep.
All right, so does everybody agree you know how to do this experiment? The next experiment that you're going to do, you are going to look at the diffusion of a solid in a solid. What you're going to do is take 3 different dyes and put them on an agar plate. This is methylene blue. You're going to work with acetocarmine, methylene blue, and potassium permanganate. What you do is you take a pinch and you put it down, if you can, as much as possible in one spot. I would get all of these started at the same time and as close as possible. So when you do your timing of 10 minutes, 20 minutes, 30 minutes, you only have to watch the stopwatch for one thing. Now what you do, you take one of these China marker, label them all. I would label on the bottom because you're going to be measuring from the bottom anyway. It makes it easier. You're going to record how wide that spot is. So I would take the ruler and I would measure and I would say, oh, right now our starting point this is 11 millimeters wide. After 10 minutes, I'm going to measure it again and see if it's gotten bigger. Say it got to 13 millimeters. What's the difference between 13 and 11? 2. So you have a change of 2 at that point. What you're going to do again is graph the change. Are you with me? Okay. And this tells you how I want you to graph it. This time, it's going to be a line graph, not a bar graph like the last one. And it should have 3 lines, 1 for each die. It tells you what to put on the X, what to put on the Y. And so that is the second experiment. The third one, you're going to take a microscope slide. You're going to put a plant, you're going to put a drop of water, and then a cover slip. What I did, I put my plant down, I put some water from the container, and I put a cover slip, just like this. And I put enough water that it actually spilled out a little bit on each side of the cover slip. That's your goal, that's important. What you're going to do then is you're going to take this, put it under your microscope, and focus it. And get it really good in focus. And one person will be watching it. One person will be getting ready to do that. All right? This, if it works, it will work really fast. In less than 2 minutes, the change will happen. So what you're going to do, take strong salt solution, take it back to your microscope. I would not take this out of the microscope. Once you get it in focus, don't screw with it. You're going to have one person looking, the other person is going to take the strong salt solution, and they'll put a drop right here. Now remember, the water was spilling out here, and you put a drop there, and immediately they're going to put a piece of paper towel on the other side. And the person who's looking through the microscope will see what happens as the paper towel sucks the water up. And when it does, this water molecule pulls on this one, pulls on this one, pulls on this one. We call that what? I'm looking for a C word. Cohesion, exactly. And what happens is it'll pull that strong salt water underneath across the cells of the plant while you're looking at it. And if it works, what you're going to see, you'll start with all the cell parts all spread out real nice. But what will happen is that as the salt flows across it, the water is very salty. We agree, right? Yeah. Salt water. So water always wants to do what? It follows salt. It dilutes things. So the water will flow out of the cell into this hypertonic salt solution, trying to dilute it. And what that means is the inside of the cell will start to shrivel. What's the C word for that? Louder. Crenate, exactly. Very good. It'll crenate. Now, if it goes far enough, you could actually peel the membrane away from the cell wall. When you do that, it's attached in a few places, so you have to rip it. If you rip, if you lyse, if you break the plasma membrane, like you see this whole cell's going like that, like they peeled the top off the can of tuna, right? If you lyse the plasma membrane, plasmolysis, you might be able to come back from crenation without dying. You will definitely kill the cell if you punch holes in the membrane. Good? Good? All right, so there we go. Now, we've talked about all three. And I want you guys to do it. I will be walking around taping as you do stuff. Then we'll edit this together in a video we throw up on YouTube for that other class. Any questions? All right, move on.